Welcome to the Dr. Bergson's Best Health Radio Show. And what we try to do is get really agile thinkers on the show. So I had Dr. Peter McCullough on just a little bit ago, and actually last weekend before I left for Florida, he had me on his show, and he lamented, why aren't there more female physicians in this scientific COVID space? So one of the people he gave me the name of, who's kindly going to be on the show this morning, is very exciting because she's a gastroenterologist and hematologist, and she has been looking at the biome and COVID. So today's show is about the biome. It's about its relation to COVID. And also because she has been the principal investigator on so many clinical trials, and she's opened up Ventura clinical trials. You're going to hear a little bit about her in just a moment. So she's really been in the peer review trial space. So she's going to talk about how frustrating it is today that we don't know what science to believe, what scientists or physicians to believe, if we should believe her or you should believe me. And that's going to be the topic of the show. So it's going to be biomes, COVID, and the reality of science. So, Hippocrates said health starts in the gut, and we often refer to the gut as the mothership of health. And if you just go to the free NIH PubMed.gov, which you can all go to, and I recommend that you do when you want to look up information. And if you put in biome and COVID, there are pages and pages of studies that come up. In fact, our guest today has published one of these papers. There's probiotics and COVID. Is there a link? Gut microbiota and COVID, a possible link. Alterations in gut microbiome. And perhaps maybe one of the reasons that the senior citizens have been so vulnerable to COVID, as we know that when you age, your biome becomes less diverse and thus less healthy. So we've got the expert on the biome and COVID. Let me give you a little bit about her background. Of course, you want to know her name, which is beautiful. It's Dr. Sabine Hazan-Steinberg. So she's the best of all worlds. She has 22 years experience in clinical trials. Let me tell you, I have one peer review original research study published and then a non-peer review. And that one study took me 3 to 4 years. So I can appreciate her elbow grease. So she's the founder and CEO of a group that does peer review trials called the Ventura Clinical Trials. She also is, as I said, and I'm going to say it again, a gastroenterologist, hepatologist in internal medicine. I think I said hematologist before. She's past chief of medicine at the Community Memorial Hospital, and she's had numerous very big awards. Like she was one of the first women to work in gastroenterology, and some of the fellows that she worked in. She's published numerous articles in magazines that are very difficult to get into, like the Journal of Duval County Medical Association and Gastroenterology. She's won several awards, like the Best Fellow Scientific Presentation and Dean's Research Award that was awarded by the University of Florida. So she's got 3 licenses in 3 different states, one in Florida where I now go work 1 week a month. And what she likes to do is be a thinker and think outside the box. And thus we have invited her to the show today. So I just want one more to get this clear. She got her medical diploma at Concordia University in Canada. Boy, Canadians are also gorgeous, and so is our guest today. She has all these different publications, and we're going to have her be on the show right now. So she's in a hotel room because she's on a tour with one of her kids for college. So she kindly had to close all the curtains and make sure the light was right so that we could see her and hear her. So welcome to the show, Dr. Sabine Hazan.
Thank you. Thank you very much. And thank you for having me. Concordia was where I did my bachelor, and I actually did science and nutrition with a minor. But Dalhousie, yes, I'm a Canadian through and through, went to school and love Canada. They teach you to think as doctors, and that was one of the beauty of being a physician in Canada.
All the men I've been in love with have been doctors from Canada. So they're really smart, and they're also gorgeous.
Yes, they are, except I married a Texan, so I couldn't find that Canadian doctor.
That's so funny. So Dr. McCullough said you were the physician to get on the show because of your knowledge of the biome and COVID. So can you explain a little bit for people? And now we have a lot of medical doctors and pharmacists that listen to the show, but we also have a lot of smart patients who are maybe walking or in the bathtub, so it's a combo of people. But if you could give a description of the biome and then your understanding in your paper on COVID and the biome.
So I think for me, it kind of goes back. I'm the kind of woman that, if I'm told I can't do something, then I do everything in my power to say, watch me, right? And so ever since I was little, people would say, well, you're not going to be a doctor. And I would say, well, watch me. You're not going to be a GI, watch me. And I think what happened is I embarked in the clinical trial world, doing a lot of clinical trials for a bacteria called Clostridium difficile, which, we had a hard time treating it. And only when we started doing fecal transplant or microbiota transplant, in other words, transplanting microbes into the colon of people, we started noticing we were fixing C. diff, right? And this bacteria. And back in the days when I started with this bacteria, and as a fellow, I remember treating these patients, we used to think, well, this bacteria is contagious from doctors and nurses giving it to patients. But what we've come to realize is actually it wasn't true, that we only get this bacteria because we kill our diversity in the gut. And that was kind of my path of discovery with clinical trials. And then from clinical trials, opening a genetic sequencing lab that made me understand Clostridium difficile a little bit better. So if you remember, 25 years ago, we were giving antibiotics for everything, right? And then it became biologics for everything. And I remember we started in GI with Remicade, right? For inflammatory bowel disease. So that was the beginning. And then pretty soon I started getting clinical trials for psoriasis because I had the experience with Remicade in Crohn's disease. And then I would get it for, and a Remicade-like medication, right?
Can you explain what Remicade did and why they were prescribed for patients with these autoimmune inflammatory gut disease?
So Remicades are biologics, right? They're basically medications that go and stop the reproduction of what creates the disease, whether it's Crohn's disease, to stop the inflammation. So it creates kind of a Band-Aid. So, and of course, when you see people taking it and they stop, then they get their diseases back, right? Because you no longer have that Band-Aid that's created in the body. So Remicade biologics started happening for, Crohn's, rheumatoid arthritis, psoriasis. And I would go to these clinical trials and realize, wow, we're using the same thought process, right? And then it became monoclonal antibodies, same kind of thing for Alzheimer's, Parkinson's, all these, medications start taking on a league of their own. Because really, if you think about it, what is creating all that? The person with Crohn's is very similar to the person that has arthritis. And in fact, the person that has psoriasis also has psoriatic arthritis, right? So things are linked up, right? It's not just a one disease. It's one disease leads to multiple. I always say an action leads to a reaction, right? So it's a domino effect. You start with one thing and then you end up with another thing. What happened is then we entered the fecal material in a capsule business, which I started seeing on my desk. Clinical trials from, fecal material from Korea as a clinical trial and, other companies. And I said, well, wait, we're in the microbiome business and we have no idea what we're doing. And that's really taking pharma to another level, right? Because then that's...
So why don't we just give a description of fecal transplant?
Yes.
For anyone who might not understand what that is. And if you might also just address... I know that sometimes giving healthy microbes from a healthy person to an unhealthy person is good. Is there really an increased incidence of autoimmune disease in some of those people? So if you'll address all that, please.
Sure. So I think so what happened to us in gastroenterology is we were, and especially for me anyways, this has been my path, right? So of discovery, I was doing clinical trials to help patients with C. diff. because it was much easier to give them a pill, an antibiotic, than having to find a donor that is healthy and having to test that donor to make sure the donor doesn't have HIV, hepatitis C, different bugs in the stools, right? I mean, it's a long process we do when we test these donors. We have to do,
donors for healthy fecal material.
For healthy fecal material. So when we entered the world and following, the brave, courageous work of Dr. Tom Barodi in Australia, who basically, was also following Dr. Einsman, who did 2 cases for C. diff, in the 60s, I think it was. So there were cases, some doctors did, microbiota transplant and showed us as young fellows at the time, 20, 25 years ago, that there was a role in microbiota transplant, right? And that role opened our eyes to the world of the microbiome, right? And so
let me just summarize this. You're saying that you were starting to treat people with healthy stool material from a healthy person and put it in someone who had disease in an unhealthy person and get improvement. And that led you to look at the importance of microbial life, which are bacteria, viruses, and fungi in the gut.
Yes, it was more of clinical trials failed. I would do microbiota transplant. And so every time a clinical trial would fail for C. diff, and I've done many, I would put these patients for microbiota transplant, and 92 to 99% of the time, the patients would do great. I would never see the patient again. In fact, I'm following some of these patients 16 years later, and they're fine. They have not had a relapse of C. diff. And some of them are young people. But at the same time, my colleagues and I, there's a group that we formed called the Biome Squad. And they're all doctors from all over the country, and actually the world. We have a doctor from China, that when we started collaborating together, we started seeing different things were changing, right? Myself, I had a case with Alzheimer's that when I did that had C. diff, and when I did microbiota transplant, the patient started remembering his daughter's date of birth. So what happens when that happens? And then Colleen Kelly at Brown University noticed that she had 2 alopecia areata patients that were bald, which is from an autoimmune process, right? She noticed that they started growing hair after the transplant. So what grows hair?
After the fecal transplant.
After the fecal transplant. All she did was take microbes from a healthy donor, probably one that has a lot of hair, and transplanted into a person that has 2 people that had an autoimmune process of alopecia areata, and they started growing their hair.
Did they grow their hair all back, or did they just grow a little fuzz, or what was it? Like?
The case
this is a big issue for many people.
Yes. So the cases are published in the American College of Gastro Journal, and you will see it's an abstract. There's a clear picture of basically a patient before and after, and you would see they're bald, and then they started growing the hair, generalized hair. So this is not like, patches. And it's not only alopecia areata, it's also, Sahil Khanna at Mayo Clinic noticed chronic urinary tract infections started improving. Of course, it's all these little cases here and there, not uniform, and we've not been able to reproduce that data until we start doing a lot of clinical trials to see who does it work for and who doesn't it work for.
Who does fecal transplant work for and who doesn't?
Exactly. And why doesn't it work for? If you look at Dr. Adams' study from, Dr. Adams did a study on fecal transplant with autistic children. 50% of kids had reversal of their neurological problems with autism. So
how quick did that happen?
It was not quick. It was a 2-year study, and then they started noticing. And the paper, got a lot of attention. But
so do you only do the transplant once, or over those 2 years, do you do it?
So he has a whole protocol that he follows. We all have our different protocols, right? He has a whole protocol that he continuously has transplants. I will say that 50% worked for, but the other 50 didn't. So that's my interest. My interest is understanding Why did the 50% fail? And why did the 50% succeed? Because at the same time that we're doing and playing with all these microbes, we run a risk of causing disease. The same thing that grows hair and changes an autoimmune process can also create an autoimmune process. And that's the big danger. We've definitely seen cases of rheumatoid arthritis after microbiota transplant. There's been cases of 2 deaths with microbiota transplant. So this is not, and I think we're up to 4 deaths.
Were those in elderly people or in who were the?
No, they were immunosuppressed. So, we have to be careful. I think, before we sell anything, we have to be careful and we have to do due diligence, right? And so that's what basically brought me to this field, from clinical trials to understanding the microbiome and opening my own genetic sequencing lab. Because what was happening was when I was sending my stools of before and after, example, the patient with Alzheimer's, I had stools before and after, I sent them to different labs and I got completely different results. It wasn't even the same patients. It didn't even match. We have the same way that we have our fingerprints are unique to us. Our microbiome is unique to us. So when I look at my stools today, and let's say I take vitamin C for a month, and I look at my stools in a month, I'm going to notice the same fingerprint, but a few microbes will have changed or shifted. That's the beauty of a validated, verified, and reproducible assay. And we don't have that right now. There is no assay in the world that is looking at the genetic sequence of the microbiome. So
I thought the GI map was looking at the genetic sequence.
It's not validated. It's not reproducible. It gives you a group of microbes. However, you cannot compare it to others. Why? Because we're all different. You and I are completely different. What your microbiome that is good for you, it may not be the same microbiome that's good for me.
So how do you take probiotics then, if you know that? How do you take probiotics?
And that's the challenge. So do I take probiotics? So I know what I started off with. I'm fairly healthy. I don't really, overdo of anything. I don't take any probiotics that are in a capsule. Because even though so we've tested in our lab, but also there's data out there that shows that 16 out of 17 probiotics, there was a paper that showed that 16 out of 17 probiotics out there are not what it says on the label.
But that paper was like about 20 years old, right? That's not a recent paper.
Even when we test the microbiome of probiotics in our lab of patients that come to us and say, well, look, I'm taking this probiotics, we're noticing that it's not on the label. Some labs are accredited and have what's on the label. However, the big question then becomes, is that bacteria live or dead? In other words, probiotics, you're supposed to mimic the gut, right? The gut is anaerobic bacteria. Remember, it doesn't breathe oxygen. So if you're giving it microbes that are breathing in, that are exposed to oxygen, you've just killed the microbes. And then you're essentially having dead bacteria in your gut. What is that doing to your gut? And that's the $1 million question. And also,
what about all the positive? There's such a huge body of positive research for this therapeutic efficacy of probiotics. Can you comment on
certain probiotics? Absolutely. If they're done in an accredited lab, if they're done under a clinical trial, if there was oversight, absolutely. If you look at the ones that have been undergoing clinical trials, and I'm talking VSL3, and I forget the other one, Biome, those were looked at, and actually they looked at the clinical efficacy of them. And, for the most part, they did more good than harm. However, what do I like as probiotics? I like the natural stuff. I like the yogurt with the probiotics. I like the kefir with the probiotics. That's what I like. I like something that came, from a farm that is natural as much as possible because, it comes from the planet.
Have you heard of one of my favorites is
Yeah.
umbashi plum paste. There's a lot of literature on fermented plums that
Yes.
give you natural, and it's called UMEBOSHI. You could do a half a teaspoon twice a day. You can get it on Amazon, shipped to your house. And I also make sushi, vegetarian sushi, and I put it in the center, and it gives that umagi, that incredible flavor. So I don't know. Have you ever? I love that.
Yeah, you know what would be nice is actually testing your stools before and after. Take a break from all that and then give me your stools before and after, and I'll tell you if actually this is good for you. But I've tested a farmer, his microbiome, who basically eats a ton of his yogurts, and he was married. He's married to a woman who got COVID, and he never caught COVID. There's actually a YouTube video of Dr. Hazen and the farmer. And he'll say in the video, I took my wife's saliva, I put it on my eyes, I kissed her, I never got COVID. And he even jokes and says, am I that useless that COVID doesn't even want to enter my body? And so that's the premise, right? That's what made us start thinking, maybe there is something to that. Why are the farmers, I work in an area that is highly populated by farmers. And
you're in Ventura, California,
I'm in
right?
Ventura, California, and there's a lot of farmers around us. And surprisingly, we've not really seen them, in the clinic. So for COVID, and I run a bunch of clinical trials for COVID, I've not seen my farmers. Usually they're the first ones to come in. So there's something about the herb. There's something about natural products and natural, microbes that is really amazing.
So tell us how you got the paper that you did on the biome and COVID, and what are your...
It's not published yet.
So it's coming out soon. That's right. So it's got the magical bacteria that you can't mention yet. It's like the drug that shall not be named. But tell us about that.
So what happened to me is when COVID hit, so remember I'm a GI doctor. I do microbiota transplant in hopes of finding cures for multiple diseases. And then I opened I have a clinical research center. And then from there, that does a lot of clinical trials for pharmaceutical companies and a lot of products, even natural products like Nestle. We did an herb for them 1 year. And then I opened the genetic sequencing lab to understand the microbiome. The first thing when COVID hit so I have 9 doctors in the family. My husband's a cardiologist. My sister's the top, brought ivermectin to the market as a clinical research doctor. I have a top dermatologist in New York as my sister. So we're 9 doctors. And I'm the oldest. So the first thing I did was, wait a minute, I better figure COVID out because my siblings and my husband is on the front line. And if you remember when COVID hit, we were left with no mask. We were on our own. We were basically like, okay, go to war, but we're not giving you any guns. So I had to figure it out. So I started calling a lot of my friends that were on the front line in Italy. A lot of my friends, I have one doctor that I'm associated with in China to try to figure out what was working. And the first thing I realized was I own a genetic sequencing lab, and the virus sits on the ACE2 receptors. So the first thing I need to do is look at the ACE2 at the stools, because where's the biggest body of microbes? In the stools.
And the gut wall, the enterocytes express ACE2 receptors.
All over. So essentially to me, I would be able to find it in the stools, right? And I told my scientist and drove him nuts. I drive my scientist nuts all the time because I go from autism to Crohn's disease to Parkinson's. Like today we're looking at MS. And then when COVID hit, I felt like we need to look at COVID. So I said, stop everything. We're looking at COVID. And he goes, we're not going to find it in the stools. And he courageously took on the task to look.
Who's he?
My scientist, Dr. Papuzzi.
Okay.
So Andreas Papuzzi was my scientist that helped me create the assay that basically allowed us to look for microbes, etc. And so when we started collecting stools, I'm friends with a lot of doctors in Atlanta, one in particular, Dr. Alan Miller. And I said, Alan, I need stools. I'm sending you kits. Get me patients with stools. So the first protocol we wrote was actually COVID-19 and the microbiome, because I wanted to see, are we able to see it in the stools? Because I felt that even though you don't have it in the respiratory tract and the upper respiratory, if it's in your gut, it's going to start creating havoc in your gut. And knowing what I know of the microbiome so far, having looked at 42 clinical trials on the microbiome and disease, I felt that as a clinical trial, I felt I was the most equipped to look at the microbiome differences that would happen in COVID patients, right? And so, and again, because we have this validated assay, we had the possibility to look at that. So I collected stools from patients with COVID, and the first 8 patients we tested that were positive nasal swab ended up having COVID whole genome in the stools. And I'm going to explain that because a lot of people think, well, when you find COVID, it's just a small segment like the PCR, right? Whole genome is looking at the whole entire virus. It's like basically having a parasite in your gut and you're seeing the whole parasite floating, right? And so it was the ability to look at the whole region and not just one copy of the virus, but thousands of copies of this virus entirely from A to Z, the whole region, with all the spike proteins, with all the mutations. So what we noticed is there were like 33 mutations in the regions. And then we noticed that 5 out of the 8 specimens that we got from different sides of the country, some from Atlanta, some from California, were all different. One from
Philadelphia. This is fecal material from all over the United States.
But yes, fecal material that is not just sent as fecal material, because when we give them these kits, they're actually a piece of specimen of stools that is in a gel that basically freezes the stools. So in other words, it's no longer feces, it's genetic material. It's a fingerprint on a carbon copy, essentially, that we then take in our lab and we basically, put tags on it. We sequence it, we library prep it, we put it into the machine, and then it goes into the cloud and essentially reassembles into what we find, right? So when we look for COVID, we have to remove all the bacteria, we have to remove all the viruses, the other viruses, and we have to zone in directly on COVID to look for COVID. So it's a very complex procedure, very time consuming, it's a 2 weeks work at least. And so, my scientists basically did the work and we found it. And then we discovered that, well, if it's in the stools, what is that COVID doing to the rest of the microbiome, right? And so from that pipeline, we started taking it to another pipeline, which is...
Wait, one question, is this just in very ill people or also people who tested positive so they could have had a mild case?
So we've been collecting over 400 samples across the country to look for every sequence, right? To look for what does it look like in different people? And also what does the microbiome look like? So it's mild to moderate to severe. So in those 8 patients, we had one that was asymptomatic and had a lot of COVID in his stools. We had one patient that was a young girl, 21 years old, super healthy. So we thought, and almost died from COVID. But guess what? She had COVID in her stools for 45 days until we started treating her. So, looking at the stools tells us the story. Looking at the stools tells us, is the patient an asymptomatic carrier? And therefore, is he going to the bathroom, flushing some COVID in there, and then is the virus going airborne, right? So I think we need to look at that. I think that's a necessity. I think China started doing anal swabs actually because they saw the importance of looking at the stools. But more important than looking at the stools and finding the virus is looking at the stools and what is that doing to your microbiome, and especially those long haulers. It's all a bacterial alteration. It's because the virus entered and occupied ACE2 receptors that normally bacteria is surrounding or protecting, right? So essentially that was the path of discovery. So we published that paper where we basically found COVID-19 in the stools. And now when we took those 8 samples and we added another 32 samples, so we have 40 samples of patients, we looked at their microbiome. And what we noticed is essentially there is definitely a group of microbes that is altered in COVID. But specifically, there is one that I'm going to be publishing that it's going to be revolutionary, and hopefully it'll get the right light for the paper.
When is it coming out, and which journal is it coming out in?
We're just submitting it, so we don't know yet. We're in the process. Writing papers.
Yes, I do know.
Data. You heard.
I'm trying to write an FDA monograph right now. Yes, I do know.
And it's time. We in 1 year, my team and I have put 5 INDs, individual.
So an IND is what you do when you want to start a research project.
Correct. We've put 5 at the FDA level. So this is like, 22 pounds of regulatory documents. We've had to hire IRBs, regulatory boards.
Right. That's something that you have to make sure that the patient's all going to be treated right. And it's the protective regulatory pathway. Right.
And then we had to create a electronic consent because, we're taking care of these patients from across the country. So we have to send them an email. We have to coordinate nurses going to their houses, collect the stools, collect everything. So it's a very challenging protocols. So when but
are you saying that this bacteria that you can't mention yet, it's protective against COVID, or it is the opposite?
So we believe that it could be a susceptibility marker. So in other words,
if you have to show, you might get COVID more and your incidence might be more severe.
Correct. And we're trying to basically show that what we all know as physicians, immunity is in the gut, right? But wouldn't it be amazing if there was a formula that equals immunity in your gut to begin with? And that's what I want to find. That's what I want to be. Which
are the bacteria that you think are the most protective?
So honestly, all this research is very new. So I'm not going to commit myself to anything, because I think as scientists, when we start saying something that is hypothetical, we put ourselves, in a bad light. I think, I like to quote Albert Einstein, who said, it's all research. If we knew what we were doing, it wouldn't be called research.
But can I ask you like a few bugs like Pseudomonas aeruginosa or Akkermansia muciniphila? Can you comment on those a little bit?
Let me tell you about C. diff. Let's go back to C. diff.
Okay.
So forever, everybody thought C. diff is a bad bug, right? It kills people, causes diarrhea. But guess what? Guess what was the first microbe I discovered in my gut? C. diff. Now, why have I never gotten C. diff toxin? Why hasn't C. diff ever secreted toxins for me to be symptomatic?
But aren't there some pathogenic strains and some non-pathogenic strains?
So I believe that in the world of microbiome, as long as things are in balance, you're fine, right? I mean, it's like, some people are, it's almost, the balance is really what is the key. I think once C. diff is out of balance, in other words, it's overgrown and you don't have enough of the good stuff to suppress it, then you allow it to flourish, and then it's going to come out and kill the host, essentially. And I have that discussion with Howard Young at the NIH, because he said, doesn't make sense. Why would a bacteria kill the host? And I say, well, what is the process of dying? The process of dying is our microbes in our guts are decomposing the bodies and putting us back into the earth, right? So the microbes are still alive and well. Think about it. The microbes you came with as a child that grew in your body are essentially there to take over your body. So the microbiome
when you're in the casket?
Yes. When you decompose, that decomposition, what happens? It's the microbes from the earth and your body that just decompose your body.
So I used to listen to this gentleman, Dr. Churaskin. He was a dentist, and he used to do labs for married people. And he'd say, if you live together long enough, your labs start looking alike, your cholesterol, triglyceride, et cetera. So are you saying that if you eat together, you live together, you make love together, do your biomes start merging together? And are you attracted to people? Is that at all, do you think, based on your signature, based on who you might be attracted to?
Yes. So my husband's microbiome. So my husband and I are similar in some areas, okay, but we're very different in our approaches to different things.
He the Hazen or the Steinberg?
No, he's the Steinberg. He's
Okay.
Dr. Lon Steinberg. You've seen him on the Conrad Murray, Michael Jackson case. He was the expert that said it's like leaving a baby on the counter. So Dr. Steinberg, my husband, he's very calm. He's a guy, everybody loves him. And, I'm not as calm because I'm more, of a hurricane. If somebody tells me I can't do something, I'm going to do everything in my power to make it happen. So we were very opposite when we met because he was that calm, laid back kind of guy. And I was fiery in a way. And then as we've been married for like almost 29 years, we're very equal. He's a bit of a hurricane as well, that he's caught on to me. But when we look at our microbiomes, both of us, we're very similar. I mean, you could see we're living together. There's differences. Of course,
it's because you tend to eat the same meals. You tend to eat meals together.
I think it's the share of the microbes.
Mucous membranes. I mean, is all that part of it?
I think it's the share of the microbes. My daughter, when you look at the microbiome of my family portrait that I always show at different lectures.
That's what you've got up in a frame for guests to see. It's not pictures of your face, it's your shared family microbiome.
And you see, like my husband and my little one are almost identical. And they're identical in personalities. They're funny, they're laid back. And then me and my oldest daughter are identical. We're high strung. In fact, we butt heads. We're so identical because, she's my match. She's my mini hurricane, So, and me and my little one, we're more compatible because we're different in a way, but it adjusts us, right? So when you look at that family portrait, you could see the differences. What's amazing is when you look at the family portrait of kids with autism, that's where the answer starts showing up in the microbiome, where you see there is a microbe that's overgrown, overtaking the space of the gut.
Plume. They call them kind of toxic plumes.
Yes, in the kid. But of course, we need to do more studies to see, is it really secreting the toxins? You've read the data from Dr. Feingold on the sulfovibrio, right? So we actually found kids that had the sulfovibrio in their stools, comparing them to the family members that didn't have the sulfovibrio. We found kids that had Clostridium perfringens, which was also a Dr. Sidney Feingold. So Dr. Feingold, when I went to him and said, I improved a patient with Alzheimer's who could remember his daughter. What did I do? He goes, you're looking at the microbiome. He gave me a paper and he said, when you open your genetic sequencing lab, you're going to find this bacteria that I've cultured for years, and you're going to put it in a safe, and you're not going to look at it. And when you find the group of bacteria that he identified as being Alzheimer's, you're going to validate my work. And so he passed away, gave me all his patents, all his books. He signed them to me. And, I felt like, tag, you're it. It was like divine intervention. And then I connected with Dr. Tom Barodi from, the pioneer of fecal transplant. And I said, Dr. Feingold and you were involved in a patent, and he told me about Alzheimer's, et cetera. And he said, so Dr. Barodi said to me, yeah, well, it's time to continue the work and continue the path, because it's an amazing path of understanding the microbiome.
Have you seen a link, a certain signature of people who get cancer with their microbiome?
We're looking at that. We are definitely, well, I shouldn't say definitely, we are seeing some things, but the question becomes, is the cancer is it cause and effect, right? Is it because you've got all these microbes that are out of whack that you got cancer? Or is it the microbes are out of whack triggering a cancer, right? So it's like, which one came first, the chicken or the egg? Kind of with COVID, what we discovered is we don't know. We don't know if people started off with a group of microbes that were dropped and therefore were susceptible to catching COVID, or COVID entered the bowels and therefore destroyed those microbes, therefore causing the disease. And so that's the $1 million question.
Have you been looking at all about the early intervention of the effects like of ivermectin or hydroxychloroquine on the biome?
Yes. So when I started my clinical trials and the first 3 INDs that I put out. So remember, I was on a path to help my family and myself. So I needed something. So the first thing I realized was vitamin D in Italy was doing great. So that was my first, vitamin on my protocol that I submitted to the FDA. So it was vitamin C, vitamin D, and zinc, because zinc blocks the ACE2 receptors. Vitamin C, I noticed that if they gave IV infusions of vitamin C, people were doing great.
Right there, that first paper in China, sorry.
Yeah, that first paper in China. So I looked at the data from China, the data from Italy with vitamin D, the data from the zinc, and I said, vitamin C, D, and zinc, perfectly safe. Nothing wrong with telling patients to take that. And 2 hydroxychloroquine.
Of course, the government didn't do that. I'm sorry, go ahead.
Well, then I did 2 hydroxychloroquine, and I know that the half-life of hydroxychloroquine is, 29 days. So I said, well, I don't think we need to give that much. What if we just give every 3 months 2 pills of hydroxy and vitamins? So I did that. And then, of course, it all went south. And then I also looked at Dr. Raoul's protocol from France with hydroxychloroquine. And was a crazy story, but hydroxychloroquine, azithromycin, zinc.
That was the Marseille study, right?
Yes.
Right.
And it was HAZ, the first 3 letters of my last name. So I'm like, divine intervention. I mean,
you were meant to do this research.
I'm like, okay, I'm going to try the trial, because if we don't look and we don't do the placebo-controlled trials with the FDA watching, we're never going to know, right? And then that trial went sour. I mean, I couldn't recruit. I couldn't advertise. Every time I put a post on social media, it's getting destroyed. There's trolls like crazy. I'm getting attacked. So I said, you know what? And I'm partners with Dr. Barotti on this, because we said, well, you know what? We're going to create protocols and see what happens. And then we decided, well, we're going to start with ivermectin, doxycycline, and zinc, and vitamin C and D. And so we pushed that one through to the FDA. My interest in those trials was really not to bring a product to market. If I bring a product to market with that, great. But that wasn't my interest. My interest is exactly what you said, looking at the microbiome. What do those products do to the microbiome? And frankly, we should be starting a world of looking at the microbiome. Every pharmaceutical company that is putting a drug out there should start asking the question, what is that doing to the microbiome, short-term and long-term? Obviously, yes, penicillin, you're going to need it for if you have a pneumonia. But then maybe you need the probiotics with it. That is the right probiotics.
Right. It's like giving Flagyl for bacterial vaginosis over and over again to a young woman. Right.
So you got to balance. It's all about balances. So it's a domino effect. What we learned from C. diff is you kill one microbe, which is strep pneumonia, for example, but then you're killing off all the clostridiums, and you're killing off all the bifidobacteria and the bacillobacteriums and all the what we think is the good diversity, good stuff. And then C. diff comes up. And then, of course, it's going to start secreting its toxins.
Because the balance is lost, and then the pathogen can create havoc.
I explain this very simply because I'm a gardener. If you have a field and you have, your beautiful flowers, roses, blueberries, and you have a diverse garden, the animals are not going to come and eat your pumpkin that you're starting to grow, right? Because it's surrounded by all that. But if you start destroying little by little your garden, and you're left with one pumpkin that's trying to grow, and you've got a lot of weeds around it, well, the pumpkin's going to be eaten by the animals, and then the weeds are going to start spreading through your garden. It's the same principle here. The bacteria, if you're allowing it to grow in your gut, and not counteracting it with diverse microbes, you've lost.
So is there any trick that you do now that you know all this with that way that you eat to maintain a healthier, diverse biome?
I'm diverse. I like diversity. So I always tell everybody, diversity is health. Loss of diversity is disease. Think about it. The sterile environment that we're living in, that's not diverse. That's a sterile environment. You're living in a box. Can you grow an apple tree in a glass container? No. The apple tree to grow needs microbes needs light, needs sun, needs everything. You cannot grow anything in a glass container. So the same thing as a human being, I expose myself. I really think the reason Dr. Feingold lived to 97 years old is because he played with all these microbes. Immunity is something you build. Immunity is accumulation of microbes. If you've got an over, if you have, and there's actually, you have to think of a door. And if you've got compacted microbes in that door, nothing passes through that door. But if you've got like just, a bunch of Clostridiums here, a bunch of Bifidobacter here, but there's a lot of space, things are going to penetrate. That's your leaky gut, essentially. Things penetrate when we don't have diversity. So diversity is very important. How do you get diversity? You get back into the earth. You get back into playing with dirt and mud and being out there, not being so careful about cleanliness, etc. Because I think the more we sterilize, the more we sterilize our guts. So we can live in a sterile environment as sterile individuals, right? And as long as you stay in that box, you are fine. But the world is not that box. Once you leave that box and you go to Thailand or Mexico, you're going to get sick. Because you didn't build up
Chicago or Manhattan.
That's it.
They're saying now that if you live in different cities, there's signatures of people who travel on subways and signatures of people who live near refineries, and it all affects. So the biome is kind of the spiritual leaders would always say we're all one.
Yes,
it's funny because your biome becomes a little bit one with the person you live with, your neighborhood, the town. It's so interesting how there is a unicity of this signature.
And I told this to my husband this morning. I said, I bet you the New York attitude is probably from their microbiome, and the Florida attitude is their microbiome, Alabama, California. You know how every different state has different.
God, maybe we could treat racism by giving them a fecal transplant.
Well, you know what? So what's interesting, and I really believe that once we start understanding the simple principle, diversity equals health, loss of diversity equals disease. You can apply it to the whole planet. Once you lose a race, once you lose a gender, humanity is not going to survive. So we need to understand that the planet as a whole needs to unite. And that's really the path of the microbiome, really.
So do you recommend like going to an Indian store and buying some things you never bought before and going to an Asian store and doing that kind of thing, eat mushrooms you've never eaten before? It's just diversity of all the different things that a typical grocery store.
I think slowly.
Translate it.
I think slowly. I think the mistake that people do is they change their diets completely. They go from what they used to eat, bagels and cream cheese and lox, to, Mexican food. And then they come to the GI doctor with bloating. Well, you've changed your fingerprint microbiome you remember the microbiome is inherited from generations and generations and generations. So you're changing that diet that you were born with, your race is born with, to something different that does not necessarily equates disease. So as much as it's diversity, I think sticking to where your diet is from, in my opinion, is probably better, because that's, you see it in Japanese that move to California from Japan, and then they see the GI doctor because they start having GI problems because they
are eating much more processed foods and less.
All of it. And also, with globalization comes globesity, comes increased autism, comes increased COVID. So globalization is not necessarily the answer. I think keeping the microbes, specific to the regions and keeping them healthy is really the answer. And stepping back.
So eating foods in season where you live, that would be something.
Exactly. Yes.
Right.
You look at Europe, right? You go to France, and I can eat a whole baguette in France and I'm not going to gain a pound. I come here and I eat a little piece of bread and I've gained a pound.
Of course, the wheat is so different. They spray less chemicals, less glyphosate, et cetera. So people go, I know there was a comedy I'm watching, I think it's called Good Girls. And she goes, how come no one has gluten intolerance in Europe? And everyone in the United States has gluten intolerance, but the wheat there is different than the wheat here.
Also, same thing with, you go to a restaurant in France, in Paris, and you ask for tomatoes. they're going to say in December, they're going to say, ma'am, it's out of season. Like they look at me like you're crazy. But
so that keeps their microbiome healthier. Yes. Can you, because we could talk for about 9 hours, but I really do want you to put your toe in the microbial mud of science. What gets so difficult? There was a survey that just came out in Medscape this week where they surveyed a lot of nurses and healthcare practitioners and asked, what do you feel about the CDC, the NIH, the FDA? What the pandemic has made us not have our same faith in these institutions as before. We don't trust. So how do we know what's reliable science and what's not, especially around COVID and the biome? You're the perfect person to chat about this.
So I've been doing clinical trials with the FDA, and I always bring up to their attention certain things, that they overlooked. And I'll always remember Scott Jackson at the National Institute of Standards saying to me, well, we count on you, the doctors on the front line of research, to tell us how it is, right? I think they count on the role of doctors doing clinical trials is very, very important. Unfortunately, only 5% of doctors do clinical trials. And unfortunately, those clinical trial companies are owned by businessmen or non-physicians. So who is overlooking the research is not as, clean as I would say, or it's very difficult to find PI oversights or PIs that are really, into the studies and watching every single thing. There are a lot of doctors.
PI means principal investigator.
Principal investigators, yes. And there's a lot of doctors that I know that do clinical trials, and we do it because we want to see. What can I tell you? I mean, it's changed. Healthcare has changed. 25 years ago, we used to do clinical trials. There wasn't advertisement on television on Remicade. There wasn't advertising.
And there isn't that way in Europe. It's mostly in America. I go to Florida. It's huge because of the demographic. Why did the American Medical Association allow that?
Well, the media, I think that, that you have to ask all the lawyers in America that are controlling all that. But, obviously all these, it's difficult because we have allowed certain things that have biased research. The moment you have a politician talking about hydroxychloroquine, you've killed hydroxychloroquine.
And that paper that was published in Lancet's lost its pristine reputation. Published a lousy paper and had to rescind it. But when I mentioned hydroxychloroquine to a very smart girlfriend of mine, she said, oh, I'm worried about its adverse side effects on my heart. And I said, hey, that paper was false. It gave a really high dose. People have been taking it for years. But so people know the bad news. They don't hear the retraction or the good news.
And I think what we've done by doing that and allowing that kind of speech is we've biased research. And imagine if that's the answer. People are going to go crazy, right? Because it's a cheap solution. But I said this pandemic is going to be over when we have a cheap solution for everyone. And, I hope the vaccine is the solution. But if the vaccine is not the solution, we've killed an early treatment. And if we killed an early treatment, we're going nowhere. We have no answers.
So what do you think is the best early intervention?
I mean, I think at this point it's too early. I'm doing the clinical trials at the FDA with, placebo control. The FDA is going to see it. I wish it would go faster, but unfortunately, I'm self-funding all this. We try to get people to, invest and create all that, but the politics and everything has ruined the development of that refurbished drug or cheap solution.
Cheap solutions aren't supported by the expensive pharmaceutical train that you have to go through to get a drug on the market. So
nobody's going to promote a yogurt for healthy living or vitamin D for healthy living when it doesn't make any money and you can't really patent it, right? And that's the bottom line. But this is not a time. We're in the middle of a pandemic. We're still in the middle of it as far as I'm concerned. This virus is not going anywhere. Let's see what happens in September, October, November, right?
So we're in early June of 2021 right now.
This is the summer. It's hot. People are outside. They're happy. They're, not as stressed. Come September, October, November, let's see what happens. And then let's see what solution's going to be happening. But, I'm continuing to do my research because I want to see what it does to the microbiome. That's my focus.
Did you get the vaccine?
I'm not at liberty to tell what I do or anything because that's just my personality.
What do you see in the fecal material of people who get the vaccine? Are you looking at that?
We haven't seen it, but we're going to be looking at it.
Uh-huh.
I believe that people's freedom of choice and freedoms, I hope the vaccine is a solution. I hope, something is a solution because at this point, a lot of people have suffered, a lot of people have died, and we need to get back to our lives. We need to get back to some normalcy where we don't think about COVID anymore, right?
So what's the best thing people can do for their microbiome to protect against COVID?
Calm down their system. Calm down those little critters in their bodies. I think the hardest thing for me to do on the front line has been holding hands of patients. And I've treated hundreds, if not thousands, of patients. And let me tell you, the hardest part is holding their hand because the anxiety is so high with COVID. So calm down and know that they're going to be fine. The best thing that I've done with my patients, and I think they'll all tell you that, is giving them reassurance that they're going to be fine. And they were fine. I've lost no one. Nobody's died on my shift. Nobody's gone to the hospital. The 2 people that actually went to the hospital and didn't want to follow my protocols ended up dying at the hospital. The rest of them, but it's hard.
Can you give your protocol?
So my protocols are actually on clinicaltrials.gov. It's under Biome, and our
Can you say that slowly? Maybe spell it.
ROGENABIOME.
We'll put it in the show notes. You can go to drlindseyberkson.com. When you look at the show, go to the podcast, and it's going to be Dr. Hazen-Steinberg, and we will have that URL for you, the address, in the show notes. So if you're walking, if you're sitting by a lake and you're listening to this and you're excited and you don't want to run to your purse in the car and get a pen, it'll be in the show notes.
Yes. So they're welcome to call us. We're super busy because we can only handle so many patients. There's me and there's all my coordinators, and I can only handle so many. One of the protocols is ivermectin, doxycycline, zinc, and we'd like to finish that one. And so we encourage anybody that's in California area, Ventura, LA, because we send a nurse to them to at home with blood draw and everything, to call us. But again, we're only doing so many. So we cannot be flooded. And really, at the end of the day, it's about quality research, right?
So this is one thing I want to bring up with you. Since you've been a principal investigator on so many clinical trials, that is rare for a physician. Yes. So that means you've got one foot into the clinical trial investigative space, yes, and another foot into the clinical space. So that's a beautiful position to be able to talk about this issue that I'd like to bring up. One of the problems that I see in the methodology of clinical trials is that you usually have to just look at one constituent. It's an active constituent. And where in medicine do we ever really just use one thing to treat anybody? With AIDS, it was a cocktail. If someone comes in even with inflammatory bowel disease, we have a number of things that we do. So in 2004, the FDA did say you can start testing herbs. And in the herbal addition to a constituent, they allowed for more diversity since an herb might have many polyphenolic compounds. But the problem, so I am very much in the hormone space, and the Women's Health Initiative was the first attempt to try and have a randomized trial to prove what we thought hormones did for decades to women, which we thought was to protect them. And because of the methodology issues, it was a statistical fiasco. It derailed physicians and women away from hormone therapies for almost 20 plus years. The 19-year reanalysis 2 years ago at the San Antonio Breast Cancer Symposium, where 12 prestigious cancer societies said, look, we went back and did a 2-decade reexamination, and it was terrible. It was wrong. In the control group, they didn't even control for women who had taken estrogen, which turns out to be protective against breast cancer. In fact, it also reduces the risk of death by 44% if you do get breast cancer and in your history you took some estrogen, which nothing's ever done. So that all came from trying to have a randomized trial with hormones, and they thought they were going to prove it. So I've become very disenchanted.
Yes.
Although I know we need research, I've become very disenchanted with it. The methodology was so poor, and it was run by 40 prestigious institutions. So I don't know who to trust anymore. And what does a randomized trial show us? And it's only on one thing. So can you please chat about that? Because I see women having lost out, more dementia, more colorectal cancer, more bone. So please address this.
I've seen a lot of good drugs not make it to market because of exactly that. It worked, but then some glitch in the protocol where they messed up something and then the drug didn't make it to market. So I think, the idea of research, remember, we started research with, we're individuals. It's the art of medicine, right? We're all different. How could we possibly require all the same pill, right? So that was our foundation. That's how we learned medicine. And then essentially, medicine became a business. It stopped being an art. And when it stopped being an art and became a business, we lost medicine, in my opinion. But the problem is that those who did make it a business eventually will become the patients. And then those patients are going to need innovations and are going to need solutions. And there's not going to be anything there because we have structured the research of these products to be in these randomized clinical trials. And I think with
right, and it elbows away common sense and expense.
But also, I understand the whole look. You got to give a sugar pill to see if the sugar pill working or not, right? But in the world of COVID, where we are going to show that there is a signature microbiome in COVID patients, and that those that are asymptomatic
once they have COVID.
Right. But those asymptomatics have a high, certain microbes compared to those that are diseased. One wonders at that point, can you really compare?
You're going to put this in your new paper, right?
Yes, but can you compare two groups, right? Can you give a guy without knowing what his levels of good microbes are in the gut, the same placebo, and then another guy, the drug, but they're completely different. In other words, this guy has an amazing microbiome. He's the farmer. I can give him a sugar pill. Of course, he's going to do better because his microbiome is better, as opposed to the guy who eats donuts for a living and basically is overweight, diabetic, and I'm giving him a medication. He's not going to do as well as the sugar pill.
But you're just giving more fuel to the argument that randomized trials we shouldn't be so mesmerized by them.
Well, and especially in COVID, I think it's a mistake. We're in the pandemic. It's unethical. It's been a disaster to enroll for this study because I have to tell my patients, there's a chance you could get a placebo. Who wants to get into that trial?
Right.
They're going to go, well, you know what? I'm going to call this
not
doctor.
ethical.
I'm going to call the frontline MDs and get my drugs for free. Who wants to do a clinical trials? So again, this is not, I had a big problem with that at the beginning when I pushed out my trials, because initially it was an open label trial, because I said, well, let's see what it does. In other words, Rome is burning. Let's put a bunch of water with different pails over the fire and shut off the fire and then ask what worked. But let's not give someone a pail of water and another one an empty pail. We don't have time for that. Rome is burning. We got to shut off the fire. And I had a problem with that. So I had to fight. But unfortunately, it was a tweet. Some guy on Twitter said, unethical trial in 5 drugs in a pandemic. First of all, it wasn't 5 drugs. It was 2 drugs and 2 vitamins and a nutrient. So why are we criticizing my trial that's open label, trying to save lives as an open label? And remember, I'm paying for the research. It's my research. So it was stopped. And then they told me to do placebo. I said, I'm not giving a placebo. I'm giving vitamins. So it was basically.
So in other words, you were trying to do a randomized trial in the middle of a pandemic, but you don't want to have a group so when you have a control group in a study, you give them placebo, but it's not really ethical in a pandemic to give someone with a potentially lethal virus a placebo. So you're trying to share the intricacies of how difficult it's been to do research right now.
So hard. And so I convinced the FDA and the IRB to let me do vitamins versus, the ivermectin versus the hydroxychloroquine, azithromycin, and vitamins. Okay. So that trial is kind of, put on hold right now because of all the politics, and we couldn't recruit.
Were you starting to see any data build up on that?
Can't really say.
No. Okay.
The ivermectin, doxycycline, vitamin C, D, and zinc protocol. It is as ivermectin, doxycycline, vitamins compared to nothing. Let me tell you, I have to watch these patients on an hourly. If their oxygen drops by 4%, they're out of the study, and I treat them off label, because I'm not going to have people die on my shift to make a protocol. That's just not going to be the way it happens with me. I've been doing clinical trials for too many years that this is not a time this is not psoriasis, right? Where I'm giving a medication and I'm seeing if the medication is working with psoriasis compared to non-psoriasis, right? I mean, psoriasis, you have psoriasis or you don't, doesn't really affect your life. I mean, it does as far as quality, but it doesn't affect you staying alive. These people
We're talking about survival.
This is survival. Right. And so these are very, very difficult trials to do. And I think I'm going to shine the light with the microbiome, because we need to start understanding immunity is in the gut, and we need to understand who has this microbiome signature versus who doesn't. And maybe that's why some are doing better or not. You're not going to do a race and put a person on a wheelchair and an athlete and expect them to run a marathon at the same time. The same way, you're not going to compare a person that's healthy microbiome to a person that's an unhealthy microbiome. They can't be compared. If they can't be compared, we shouldn't be doing those kinds of trials. We should be doing open label to see what works and treat as many people as possible. The art of medicine goes hand in hand with treating patients. The oath is to treat patients. The oath is not do nothing. I didn't take my license, my medical license, to not treat.
You're meaning if an elderly person gets a diagnosis of COVID, you don't just send them home to do nothing and wait for them to get worse to then go to the ER. You want early intervention. Yes. That's what can you, with all everything you've done deep dives into the microbial life of our nooks and crannies more than most physicians. Right. Are there any 2 or 3 major things we should avoid that you have seen are damaging to a healthy microbiome? Anxiety,
a lot of coffee. Cut down on a lot of coffee.
What's a lot of coffee? Because I'm a coffee
drinker. More than one cup a day is not good.
Oy vey.
We know it causes reflux, causes GI distress. Alcohol, obviously, 1 glass of red wine improves your microbiome, but more than that is probably not a good thing.
What about the people who are now drinking so much bourbon and whiskey, almost 40, 50%?
Well, that's probably not a healthy thing or happy.
Okay, because a lot of young people, this is so trendy and in. All the commercials show that they're interesting men if they have these single malt liquors. So it's creating earlier incidences of esophageal cancer, colorectal cancer.
And so now my husband is pushing me. Here he is.
Okay, there you are. There's Dr. Steinberg. He's so cute. He is so cute.
Oh. He's very cute.
He is. I might have to go have a heart exam with him.
I know. Isn't that funny? He gets the heart and I get the fecal material. Life is just not fair for women.
Well, I have to tell you, my best friend is Janet Steinberg. So I love the last name Steinberg. So I'm very...
All right. So he's rushing me because we got to go see this.
Okay, well, thank you very much for the time. And it's nice that we ended with such a beautiful mutual biome. Okay, so if you love conversations like this, consider going to drlindseyberkson.com/membership, where I've created during the pandemic, since I wasn't running around lecturing, groups of physicians and medical docs. And then there's also a group for smart patients where we have these kind of conversations and we share data. In the morning, I go through peer-reviewed data for a few hours and post it. So if you're interested at all, please don't forget to go to drlindseyberkson.com/membership. Look at this conversation that you just got in on. And here she is in a hotel room. Thank you so much for sharing all of your work and for all the work that you do. We're looking, when you publish your paper that we're all on the edge of our seat, I'll go back into the show notes and put the link to your paper that's not published yet.
Perfect.
So that, okay, will that be great? Because I can't wait to read it. Thank you so much. I'm so glad to have had this. I never met you before, but this has been a great conversation.
Thank you so much.
Okay, be well. Good luck with college hunting.
Thank you. Bye.
Okay, bye-bye.